Good afternoon, students. Last class we concluded the class with artificial pollination. Just a quick revision for the previous topic. We started with pollination, self-pollination, cross-pollination, agents of pollination, in which you have studied entomophily, hydrophily and anemophily with the special characteristics of flowers, and last artificial pollination and its importance. Today we will study about fertilization. Look at this diagram. This diagram explains the complete process of fertilization. What is fertilization? The process of fusion of male and female gametes. Male gametes are pollen grains, female gametes egg cell. This diagram is the longitudinal section of pistil showing fertilization. Stigma, style and ovary are the parts of a pistil. Inside the ovary, an ovule present. An ovule with 8 nucleated 7-celled stage. While explaining, look into the diagram and the labeled parts. An egg cell surrounded with synergids. Keep the egg in its proper position. That is the function of synergids. Opposite to synergids or egg cell, 3 antipodal cells are present. They are nutritive in function. Middle part, a large central cell with 2 polar nuclei. This is the structure of ovule or embryo sac. During pollination, pollen grains are falling over the stigma. It begins to grow a pollen tube. Pollen tube produce enzymes which digest cells of stigma and style and create a pathway. So this is the diagram that which shows structure of pollen grain. Pollen grain is germinating on stigma and germinated pollen grain. And the B diagram that which shows a pollen tube with 2 nuclei, a generative nucleus and a tube nucleus. Later on, this generative nucleus divided it into 2 male nuclei. The function of 2 nucleus is to direct the pollen tube towards the embryo sac. Meanwhile, the generative nucleus inside the pollen tube divide to form 2 male nuclei. That is clear in this picture C. The pollen tube enters inside the embryo sac through an opening called micropyle. Micropyle is the minute opening present in this embryo sac. Through the micropyle, the pollen tube enters inside the embryo sac and it bursts and releases 2 male nuclei. One male nuclei fuses with egg cell to form zygote, and this process is called fertilization. The other male gamete fuses with polar nuclei to form endosperm, which provide nutrients for the developing embryo. This much is the process of fertilization. The zygote formed later develop it into embryo. This is the flowchart that which shows the process of fertilization in flowers. Along with that, what are the changes happens to a flower after fertilization. Landing of pollen grains on stigma. Then, a pollen tube emerges from the pollen grain. It grows down through the style to reach the ovule within the ovary. On reaching the ovule, the tip of the pollen tube bursts to release male gametes inside the ovule. The male gamete fuses with the female gamete, producing zygote. The zygote divides to form an embryo. The ovule changes and develops into a seed, and the ovary changes it into a fruit. All other parts of the flower, sepals, petals, stamens, and style, wither and fall off. Over time, the fruit ripens and releases the seeds. The seeds are dispersed by wind, water, and animals to different places. On finding optimum conditions, each seed germinates and grows it into a new plant. So this is the flow chart that which shows fertilization and post-fertilization changes in flowers. A seed is the final product of sexual reproduction. Seed develop inside the fruit and a seed is a matured ovule. Inside the seed, the future plant is packed in a dormant or inactive stage with adequate food and protective covering. Fruits are ripened ovary. Fruits are of two types, true fruits and false fruit. True fruit means the fruit which developed from an ovary. Mango is an example, it is a true fruit. Some fruits that which is not developed from the ovary, any other than ovary, any other part of the flower, develop it into a fruit, that is called false fruit. Examples are apple and pear. In some fruits, the walls of the ovary is fleshy and some of them they are dry. Fleshy fruits, example, papaya is an example. Or in case of some of them they are dry, pea and gram. What are the functions of a fruit? The fruit protects the seed from unfavorable conditions and it also helps in dispersal of seed. What is a seed? A seed is a matured ovule with a dormant embryo. Now we will learn what is the structure of a seed. This diagram that which shows a bean seed along with structure of a bean seed. A seed of bean consists of a seed coat, an embryo and cotyledon. The brown colored outer covering is called seed coat or testa. Inner to testa, a transparent membranous covering called tegmen. On removing the seed coat, white colored two cotyledons are present. In between the two cotyledons, the embryo is seen and a short curved embryonal axis. Cotyledons are attached with embryonal axis. The axis consists of two parts, an upper plumule and a lower radicle. Plumule consists of two leaf-like structures. Later it grows it into a shoot system. The part of the plant which is above the soil change and the radicle which the lower part changes it into a root system. This is the diagram that which shows the structure of a seed. In this, the structure of the seed, testa, tegmen, that is outer covering, that is called seed coat. Cotyledons, the reserved food material for the growing embryo. The embryo in between with a plumule and a radicle. Plumule, two leaf-like structure. Later on it changes it into shoot system, and the radicle during its growth changing it into root system. When the seed gets sufficient air, water, temperature, the dormant embryo becomes active and grows it into a new plant, and the process is called germination. After germination, the embryo completely develops it into a plant or a seedling. With this much explanation, we conclude this chapter. In this chapter that we studied types of reproduction, asexual reproduction in plants, different types with examples, vegetative propagation, natural and artificial methods, sexual reproduction in plants, pollination, pollinating agents, artificial pollination, fertilization and germination of seeds. Germinating seeds. For better understanding structure of seed, observe the demonstration of various parts of a bean seed. Today we will study the structure of a bean seed, that is kidney beans. These kidney beans, the shape which give that name kidney beans. A clear concave and a convex side is visible. That is why they are named it as kidney beans. So this kidney bean, the concave side, a whitish scar-shaped structure, that is the hilum. Near to that hilum, a minute opening present, that is a micropyle. Micropyle helps in exchange of gases. And the outer, this brown colored cover, that is the seed coat. The seed coat is formed of two coverings. The outer visible covering, that is the testa, and inner to that a membranous covering present, that is the tegmen. Here this is the seed coat removed, and the seed coat, it is having an outer covering, that is the testa, and inner to that a membranous covering, that is the tegmen. When we remove that outer covering, then we can see such type of a structure. The white colored part, that is the cotyledon. These are the cotyledons. Cotyledons are the reserved food material for the growing embryo. The seed coat is removed. Now open the seed cotyledons. When you open the cotyledons, you can see such type of a structure. So in between the two cotyledons, an embryo is present. This is the embryo. Embryo is having two parts, an upper plumule and a lower radicle. The plumule is having two leaf-like structure. The plumule is changing it into shoot system, and the radicle is changing it into a root system. Now this part, this is the embryo. It is separated from the seed. This embryo is having two parts, upper plumule and the lower radicle. And this is the seed, that which shows the radicle, that which is changed as in the form of a root. And this is the completely developed bean seed, and the radicle is changed as in the form of root system, and the root with root hairs, and the upper part plumule is changed as in the form of shoot system. And this is a complete seedling. So the structure of a bean seed with an outer membranous covering, that is the seed coat, it is formed of testa and tegmen, and the whitish scar, that is the hilum. Then when we remove the seed coat, inside that the two cotyledons are there. In between the cotyledons, embryo is present. The embryo is connected with the cotyledons with the help of embryonic axis. Embryo is having an upper plumule and a lower radicle. Plumule is changing it into shoot system. Radicle is changing it into a root system. This is the complete structure of a bean seed. One more, that embryo that you can see here in this one. Another one, again that in this also, that you can see the embryo that which is present here in between the cotyledons. So this is the complete structure of bean seed. I hope you understood the structure of a dicotyledonous bean seed. Observe all these parts in any of the dicotyledonous seed. Thank you.